Hi everyone, I'm Zakaria Louadi, a PhD student from the Technical University of Munich. And in today's talk, I want to go a little bit beyond protein-protein interaction network and try to explore the impact of alternative splicing. So the first section of my talk is about DIGGER, a database we published last year. So first of all, why we are doing this, why we should look at alternative splicing. So most of us are familiar with PPI networks, where we try to represent interaction between proteins as a graph. However, given an edge into network, it's not really clear which isoform are the one interaction. So we know that one gene can make a lot of isoforms that are not necessarily behaving in the same way, so not having the same interactions. So we know that at least one of them interact in this edge, and ideally we would like to try all of these combinations, but experimentally that's not very feasible. And this could be a big source of false positive in our analysis, especially that these isoforms are not necessarily co-expressed, right? So how we tackle this challenge? So first of all, let's suppose we have a known genes with alternative splicing events and some interaction into PPI. So we use domain-domain interaction to enrich the PPI, and that's basically what give you the domain that is mediating the interaction. So for example, this gene have two domains, and now for every interaction we know which domain is responsible of this interaction. We can also use residue level evidence. So this is co-reserved structure from the PDB database, and this residue can be mapped to their amino acid and eventually to their original exons. So we combine all of these evidence level of interaction, and given that we already know annotated isoforms are their domain structure, for example from the PFAM database, we can detect isoform specific interactions. So for example, this gene have two annotated transcript and one exon skipping event, and that cause the loss of this domain. So domain 1 is missing in the transcript 2. So we could eventually detect the specific interactions that are mediated by this domain. And here we compare between two isoforms, so the interactions that are shared between them and miss. So we could also do that at exon level. So we basically map all exons to their domains and their residue, and then look at the specific interaction that will be lost by this exon or are responsible of this exon. And we could do that in network level, basically, which is just an extension of the first mode. And basically you want to look at interaction between multiple isoforms. So as I said, DIGGER can run on multiple modes. So you could input an exon and look at exon skipping event, for example, or you could look at specific gene and compare between isoforms. So this is useful when you have an isoform switch event and you will look at the exact impact of such an event into PPI. And you could also run it on network level analysis, which is I'm going to explain it more into the coming slides. So this is how it looks like. So this is an example screenshot from DIGGER. So you can see here two transcripts from the same genes, and one of them is missing this domain over here, and you could see all interactions that are affected. So this is here we visualize only one interaction and two missing domain. So we also provide a score that basically tell you how much confident we are of these isoform specific interactions. So the network level analysis mode is designed to generate a condition specific network or PPI that consider that not all isoforms are same, right? So you have a list of isoform and you only want to look at the interaction between these isoforms. This could be a protein complex that where you know some variant are only expressed in one condition, for example, or could be just a pathway or a module. So you start from the reference PPI. You annotate it with the domain interactions. So this is just like the previous mode. It just generalize it over all of the PPI. Then given a list, this is user list of isoform or transcript. So first of all, the obvious thing will be to filter genes that are not into list or not expressed, but we also filter domains that are missing, so they are not specific to our list. And we detect the interaction mediated by this domain. So you could visualize this using DIGGER web interface. You could also generate a weighted network. Basically, for every edge, we reweight depending on the domain annotation that are annotated. So for example, this weight will be 0, given that all domains mediated this interaction are not expressed. So this is how it looks like. Here you have the gene CD22 annotated into PPI with this interaction, but this interaction is identified as lost or missing because the specific isoforms expressed in this condition by CD22 are not able to interact or are not interact. So you could run all of these mods together. You could start from a set of isoforms, construct a condition-specific PPI, and if you have a specific interaction or a specific gene of interest, you could click on it and see all its isoforms, and even go to exon level and see which actually caused it that this edge lost or gain, depend on your condition. So my second section of my talk is more about differentially spliced analysis. How can you use DIGGER database and this knowledge to do some functional enrichment of alternative splicing events? So here we present a Python package called NEASE. So usually in your RNA-seq experiment, you have a list of spliced exons. Basically, you compare control, case, condition. You have a list of exons that are differentially used between these two conditions. And usually what people do in a lot of papers, they just go to differentially spliced genes. So from exons, they go to their genes and they try to do some pathways enrichment. But as we said, this neglects the fact that some isoforms are completely different, or they don't really share a lot of interactions, so they could have some specific interaction that are lost or gained, depending on your conditions. It can also neglect the fact that not all alternative splicing events are necessarily functional into protein level. So the ones that are affecting domain are definitely functional and affecting binding. And hopefully we can use this information now provided to do some pathway enrichment. So for example, these could be differentially spliced exons from a disease conditions, or just dysregulated exons, or like brain upregulated exons. So how can we do that? So now using DIGGER graph, what we get from a differentially spliced exons is a list of genes or edgetic genes. For every gene, we detect some affected interactions. And basically you have a pathway or a disease of interest that you want to see if this is significant, if these interactions are significantly affecting this pathway. So here we propose a hypergeometric test, and the significance of these interaction are actually dependent, of course, of the number of the interactions that are connected to the pathway, but also of the degree of the network or of the pathway itself. And that's give you a general p-value. So basically tell you if these numbers, so here 2, is significant from the overall degree of the pathway. But you also know that sometimes you have hubs that could be connected to a network just by chance, and you also want to prioritize some of these events, right? So for example, this event looks more significantly affecting this interaction, this pathway, than this gene over here. So we run also a similar test on gene level for just prioritizing to events that are affecting the pathway. So this is the overall method of NEASE. So you first run a gene set enrichment at edge level, and you do that for all of the pathways. You correct for multiple testing. Then using the Python package, you can also focus on single pathways. So for example, here pathway A, and prioritize the events. And because we are doing enrichment at the network level, so not necessary to genes will be a part of the module or the disease, or known to be a part of the disease, associated to disease. It could be a new candidate, for example, this gene 3. But because you check if it's significantly affecting a pathway, we could also prioritize or find new candidates that could be misregulated by splicing, right? So in order to evaluate that, we run that on VASDB database. So we basically extracted 2,800 exon skipping event from 11 tissues. So we extract the psi values, we run clustering, and hopefully you can see here that there is two distinguished clusters here. So this is to standardized psi values, and we try to compare to psi values across all tissues. So you can see here that there is one distinguishable cluster here in neurons and another one in muscles and heart. And basically what does tell you, these exons are upregulated in these tissues. So we wanted to check if we will find any interesting enrichment of these two sets. So first we run gene level enrichment. So this is, you go back to the gene and you try to do a Fisher test to the gene levels and see what pathway you will do. Then we compare it to NEASE enrichment, and you can see over here that using gene level enrichment you get very general terms compared to NEASE enrichment that capture a lot of muscle specific terms. This is for exons that are upregulated in muscles and heart tissues. We also saw similar things in neural tissues where we found also really good enrichment of synaptic cycles pathway. And you could also visualize that with the help of DIGGER and the Python package, so you could see the specific edges. So this is an energetic change that's caused by alternative splicings, and you could see how it affects one single pathways and how these co-regulated exons that are upregulated here in brain are affecting the pathway. You could also prioritize this event and look at the most significant ones. So we also run these on multiple other data set, but in respect of time we don't present them here. So a conclusion. So hopefully I convince you that neglecting to impact of AS may lead you to a significant bias in your network analysis. So you should definitely consider that different isoforms are not necessarily same or interacting the same. So DIGGER tried to close this gap by trying to provide visualization and analysis in different levels into impact of alternative splicings. And using our Python package, we also proved that in a lot of cases, we improve over gene level enrichment for the exact task of alternative splicing. And one reason of this could be that not all alternative splicing events are necessarily functional at the protein level. And another reason could be that we've seen that different interaction or different isoform have very different partners. So you want a method that can actually capture that and when you try to do your enrichment analysis. So to end, I wanna thank all my supervisors and everyone. I take any question if there is any. Thanks.